Welcome to the Xiuxia Exposomics webinar series. For those of you who joined for the first time, this is a webinar series organized by the grantees of the Human Health Exposure Analysis Resource to specifically focus on various topics around exposomics. And I'm Xiuxia. I'm the moderator today, and I'm from the National Institute of Environmental Health Sciences. Today is the 7th webinar in this series, and the topic is computational workflows for untargeted exposomics. We will have 2 wonderful speakers today, and each will discuss a computational workflow they use for processing and analyzing untargeted metabolomics data. So each presentation will be 20 minutes. If you have questions for the speaker, please post them in the Q&A box as they come up. We'll try to address them at the end of each presentation. So our first speaker is Dr. Oliver Fiehn. Dr. Fiehn is a professor at the UC Davis Genome Center and a director of the West Coast Metabolomics Center, University of California, Davis. I have known Dr. Fiehn for quite a few years from the Common Fund Metabolomics program, where he directs a compound identification core. So Oliver, it's great to have you. Thank you for presenting today.
Thank you for having me. Okay, welcome everyone. I want to talk about our data processing analyses on either LC-Q executive instruments or LC-Q-TOF instruments. We have, over the last 3 to 4 years, produced an automatic data processing schema that we call LC-bin-based, that is specifically targeted for large cohort studies, producing high-quality data, including exposome data. So when we talk about high-throughput metabolomics, what does it really mean? It means that, when we think about classic workflows, that in classic workflows, you would have sample by sample, you would prepare those samples manually, you would then put them into little vials in autosamplers and maybe run a 20-minute run LCMS study, and then somebody would sit on some kind of computer screen and go through studies one by one, which is taking a lot of time. The high throughput way would improve all 3 steps: the sample prep, the data acquisition, and the data processing step. So first of all, we only deal now with some 96-well plates in high throughput manner that are barcoded. We have improved the cycle times per analysis to 5 minutes, and we do everything based on cloud processing and not anymore manual data processing. So the biphasic extraction that we use for many, many years that separates lipophilic compounds from hydrophilic compounds has been improved using multi-pipettes, multi-centrifuges, and then these aliquot layers can be separated into lipophilic and hydrophilic fractions using guided plates that allow us to put these at the right spot. Then we perfected the LCMS data acquisitions. We still need to do positive and negative mode electrospray in 2 separate runs, because that's still not an option to do online positive-negative switching. We thought about all possibilities, like increasing the speed, but of course, when you increase the speed, you also increase pressure. You can use smaller particles, but then you have a lot more pressure, back pressure in the column, or you use shorter columns. So overall, we think the particle size at 1.7 micron is just fine. You can increase the speed of linear velocity without resolution. And when we tried that with maximum pressure hydrophilic interaction chromatography, we got very good resolution, also chromatographic resolution, even in high throughput. You see this here for amino acids. When we applied that for human plasma, we got 740 identified lipids and 413 identified polar compounds. And there we already got the first incidence of exposome compounds. Here you see theophylline, atlanticin in different groups. These are food compounds, of course, and very big differences and very clear identifications in hydrophilic interaction for these types of compounds. So that was already giving us the right data sets to be used in high throughput data processing. High throughput data processing combines two aspects. One is the mere data processing that you see here in an overall diagram on LC binbase, going from the user who uploads the raw LCMS data to the cloud. On the other hand, we also need a study design database. The study design database tells us, for each study, what are the species studied, what are the organs and cell type studies, and for both of those, taxonomies are available that directly, put these types of information into the right buckets. Where I myself have not seen really good informatics solutions is to categorize systematically, maybe with taxonomies again, the differences between treatments, disease groups, and time points and so on. What are the right ontologies for that? That is still, in my opinion, an open question in the field. Now, with that information, we are able to automatically and at scale do MZML conversions, peak picking, retention time alignments, looking at the MS data quality, and annotate all mass spectra, including gap filling for missing values to ensure that we have these. All these data would then be collected in the LC binbase to give us metabolome atlases so that we can interrogate studies across species, between studies of the same type of biological experiment. And we identify compounds based on both retention time, normalized retention times, as well as MS spectra. And these spectra come from massbank.us. It's a database that's hosted by my group. And all that is organized and maintained by Geert Wohlgemuth, who works with me for over 20 years. So 20 years ago, we started making a database called GC binbase for a very specific type of data acquisitions or data format. These are LICO GC time of flight mass spectral data, where LICO had clearly superior software for data processing. And we were using those processed data to detect first the internal standards to calculate retention indices, and then go stepwise, peak by peak, starting with the most abundant peaks, look for retention index windows, look for unique ions that separate the specific compounds in different regions. Then only we do MS similarity, separate the isomers, and declare a certain bin to be found. Importantly, GC binbase, as you can see here on the right-hand side in the diagram, the flowchart diagram, is also able to automatically add new compounds. So it's not needed that a user has to say, we find a new compound, but it's automatically done. However, of course, you have sometimes questionable peaks, low abundant ones, or peaks that have bad MS spectra. We automatically recognize those and only let compounds through that are found in sufficient quality and sufficiently often per biological class, enter them into binbase as a new bin. Then we reprocess all of that and export it. So the LCMS binbase worked on the same concept. And really important is we use Amazon Web Services for speed, memory, and repeatability. So we are not bound anymore by any clusters we have here at UC Davis or the memory that people would have on their workstation. We can process any type of LC MSMS file from any vendor using the original code MS-DIAL that we have rewritten for cloud computing. And we automatically, of course, associate the all MSMS spectra with the precursor masses. We merge duplicated bins that do exist because of the parallel processing. When we do a lot of samples at the same time, one sample might get a specific bin and the other one would find the same compound, so it also generating a bin, and we merge them when the study is completed. And of course, we have a lot of overhead for data management to keep the consistent database entries, enable graphic user interfaces for annotations, both also on quality metadata and comments. And I want to give you today the overview how that is working. So here, first of all, the retention alignment, you see it for a couple of compounds. We use 79 internal standards for lipidomics, and that is like anything that's lipophilic, that includes a lot of exposome compounds, and 32 internal standards for hydrophilic interaction chromatography. So that's a major break from other software that uses complete similarity alignments. So we 100% and only use internal standards for retention time alignments, and that is perfectly done because we have so many internal standards. It's much more robust. That means also it's independent of the matrix. It's much easier to quality control the alignments. And of course, now you can do studies where you can compare, for example, urine, stool, and plasma, which is very hard to do if you don't do it with internal standards alignments. These alignments are fine. They're actually better than MS-DIAL by itself, because you might get duplicate bins that are merged in MS-DIAL if there's sometimes two isomers appearing in a sample and sometimes only one. MS-DIAL is not very good in that. So then we generated graphic user interfaces, and this one is done by our senior scientist, Yunyu Li, and we call this MassWiki. MassWiki now gives you the full data set. It's a public URL. Anyone can look at it. It's not directly the administrative GUI for LC bin based, but it gives people the ability to interrogate all mass spectra. And again, for us, a peak is only a peak that has an MSMS spectra. Just MS1 ions are not going into LC bin base because without an MSMS, you cannot qualify these compounds anyway. So we have 4 types of identity searches. First of all, the classic one that you see here, that gives here the entropy score for the MSMS, where we subtract the precursor mass. And the precursor mass is an independent character that we look at here in terms of the delta mass. And if then a user would think that this is a good match, the user would click submit, and that would go to the graphic user interface for administration of LC bin base, but not automatically. There's another level for administrative review to push or annotate and reject things. Now, importantly, MassWiki has all public available MSMS spectra, both the GNPS and MassST, but also Mass Bank of North America, many other libraries, but also licensed libraries. For example, the NIST-20 is behind it. That, of course, can only be used when you have the corresponding code for having a license for NIST-20. Now, it's not only the identity search, but you can also do open search. Open search means that independent of the precursor ion, you might have a really good MSMS set. And then we see here, again, for cholic acid, a very good entropy score and the delta mass -18, which is, of course, corresponding to a water loss. We also have neutral loss and hybrid searches. Hybrid searches, similarly as they have been implemented in the NIST-MS software, and neutral loss searches that enable you to systematically look for neutral losses. So all this is on MassWiki, and basically the front end for LC-Binbase. LC-Binbase now finds sample-specific compounds. This is from the NIH Common Fund that Dr. Kui mentioned. Here we had plasma, brain, heart, muscle, and liver, and we see certain lipids that are only in brain, for example, PC44-2, or certain lipids that are only in brain and muscle, but not in liver, heart, and plasma. And that explains the beauty of cross-study and cross-matrix alignments and comparisons. More importantly, of course, we want to talk about exposome. So the first parts of exposomes are microbial lipids. Microbes, we live with them, we love them. They often come from gut. And here, for example, are specific acyl phosphorylethanolamines that are released by the small intestine into the bloodstream to act on the hypothalamus and suppress appetite. We see here the specific lysyl forms of these N-acyl phosphorylethylamines, very clear MSMS, and we see, you see here also the front end, and you see also how we report all detected and identified compounds with a splash ID. So the splash ID is a unified spectral hash key for MSMS spectra. So to enable correspondence between databases, the precursor mass, the name, the type of compound, which is a metabolite, INCHI key, SMILES, and the classifications from classifier. Then we also see other types of microbial lipids, and these are phospholipids. We found now clear evidence that phospholipids are mostly made by microbes, not by humans. And phospholipids have a very big effect on overall physiology in humans. We find many of those in plasma now, and therefore we think that they are should be categorized also as exposome compounds. Obviously, we find a lot of PFAS. PFAS are the forever chemicals. They are in fairly high concentrations in human blood. You are easily recognizing them by the accurate mass already, because that accurate mass is due to the fluorine atoms lower than the nominal mass. And in addition, of course, we would have the MSMS spectra to enable us to, identify these compounds. And then we went on compound class by compound class. We found halogenated compounds. Here are a number of them from, for example, herbicide, chlorpromide, hexachlorophene, but also medications and pharmaceutical intermediates like bromobenzophenone. And for each of those, you have not only the accurate mass, but also excellent MSMS spectra. We see a lot of household and other industrial chemicals, from benzothiazoles, which is a rubber vulcanizer, fire retardants, quaternary ammonium compounds are compounds of concern to the California EPA. California is the only state in the United States that has an independent EPA, and we named these types of compound classes as a class of concern because of the ubiquitous use and because these can be integrated into the mitochondrial membrane. We also see these exposome compounds like propylparaben from cosmetics, sunscreen agents, and disinfectants. Then we obviously see food compounds. Well, we have to say these are clear exposome compounds from ethyl sulfate, which is a classic marker for alcohol consumption, but also polyphenols like daisine and daisine sulfate, the sulfation coming from the liver, of course, but also sweeteners, then classic ones like caffeine, theophylline, and so on. So these are food compounds, and these are just examples. The biggest class of exposome compounds are clearly pharmaceutical drugs. We see hundreds of these pharmaceutical drugs in large studies, and these are all coming from two studies. One is the large-scale human longevity study with 4,000 subjects, and the other one is the TEDDY, type 1 diabetes study on environmental determinants for type 1 diabetes in about 13,000 children. So both of these are ongoing, and that's why we know that these are all originally verified detections in human plasma. We see surprisingly also a number of pesticides, like fipronil sulfone, which is a broad spectrum insecticide. Dinesh Baropal made a database called the blood exposome database. It's actually 46 times found in the blood exposome compound. So yes, we see it too, and that's with untargeted methods. Broad spectrum herbicide dicamba, fungicides, insecticides, anti-fouling pesticides, ovicides. So these are all compounds that we clearly do not want to see in human plasma, very clearly detected, unambiguously identified, and can be profiled in high throughput. Overall, we have detected and positively identified over 590 exposome compounds so far. Many of them I've shown to you, and the LC-bin base is ready to go. In addition, of course, we find compounds like oxidized lipids that are absent from libraries, for example, from Metabolon and Biocritus. This is exactly what academia do, go above and beyond what you would get as typical services from down-the-mill companies. So here you see the benefit of using academic laboratories, just to say so. And overall, then we would normalize the data, give normalized data to our collaborators and publish those. This is by random forest, the normalization that we published a number of years ago, with very small residual data errors. Overall, I would like to thank you. Large-scale, high throughput, untargeted LC methods possible, guided exposition with 96 well plates, reprocessed MS-dial, and application on human cohort studies. Thank you for your attention.
Thank you, Dr. Fiehn, for that excellent presentation. I want to remind everyone, post your questions in a QA box. I think there was one question here, is there an R package to use for MS-dial processing, specifically for GC Orbitrap data processing?
It's not needed. MS-dial can be directly downloaded from our partners in Japan and can be used directly for GC Orbitrap. So that should not be a problem. If there's a problem, please tell Hiroshi Tsugawa. So we use MS-dial for GC QTOF data. So in principle, there's no problem.
Thank you. Well, others may, type their question. So I have a question. So already you mentioned that, the high throughput SC system, LC system has pretty good resolution, right? High resolution, good resolution. I was just wondering, will you shorten the runtime? Do you risk kind of losing sensitivity over some metabolites?
No, because you lose a little bit resolution. You did, when you we had about 30% loss in chromatographic resolution, but on the other hand, you also get sharper peaks. So you get an increased signal to noise because you basically have slimmer peaks, and that's why the total number of metabolites that we detect in the short runs compared to the long runs is the same. So you lose some isomers, not many. So things that were not well separated before are then not separated at all in the 5-minute run. But overall, we don't lose coverage.
That's great to hear, because human samples are precious. So people try to get as much information as you can from limited amount of samples.
Yes.
And okay, there's another question. Okay, it's the person asked, can you talk about running untargeted exposomics to screen for unregistered contaminants?
Yes, we can identify everything that is known to the public. So it has to be in MSMS library that are publicly available, or can at least be licensed. So anything that is that doesn't have an MSMS spectrum is really hard to identify. So when we look at the EPA dashboards from Tony Williams, there's of course more compounds there, and we would love to get our hands to put onto these chemicals to put MSMS spectra to the public.
Thanks. There's another question. Says very impressive work. I'm very curious about your MMS spectra. I was wondering what the levels of these compounds were in your samples. The spectra look clean.
Yes, so the spectra are very clean because they are all generated here. I showed only Q-exactive data. Q-exactive, as the name says, is very exact. As I also explained in the to Xiuxia, sorry, to Dr. Kui, is that we have a higher signal to noise because we have less broad peaks, we have slimmer peaks, you get better signal to noise. That's why you also get better MSMS spectral quality. All these spectra are coming from human samples. These are not spectra from chemicals themselves, like the purchased chemicals, right? So they are all directly come from the samples. Is there an interface to export the spectra to a computational MS software? Yes, we are happy to, once we publish LC-base, we haven't published that yet, all the spectra, both known and unknown, will be publicly available, obviously. Everything we do is public.
And there's another question, talk about the quantitation of analyzed identified in NTA.
Yes, so quantitation is a big problem. Obviously, for good quantification, you need stable isotope intermediates. You need calibration curves. You can do a little bit by also sample dilution. That is a big problem for everyone in the field. Also, the missingness, of course. If a compound is missing, is it, well, what does it mean if it's missing? What's the value? Is it really zero, right? So that is something that we, as the field, still needs to address. Absolutely.
There's another question. I think I will try to address this question before this one, before moving to the next talk. As you probably can read there as well. You have identified many compounds. Yeah.
So we have many more unknown compounds than known compounds. So the dark matter of the exposome is still very dark. So I think at current per mode, like positive, negative, and so on, we would have about 1,000 identified and about 6,000 to 9,000 unidentified compounds. So just to give you a ratio. So about like, I would say 10%, maybe 15%. Of all peaks are identified, and the others are, despite our efforts, unidentified.
Thank you very much. I think, while we're going to move to the next talk, and people can still, post the questions there at the end of the talk. Hopefully we'll have some extra time. Or Dr. Fiehn can try to address some of those in the chat, not in the chat, but in the QA. Okay, so thank you very much. Our second speaker is Dr. Xiuxia Du. Dr. Du is a professor in the Department of Bioinformatics and Genomics at the University of North Carolina at Charlotte. Dr. Du is MPI of the North Carolina here Untargeted Lab Hub. She also leads a data analysis and tool development project in the Computational metabolomics program, where I learned a lot about her wonderful research work. Xiuxia, it's great to have you, and please go ahead.
Thank you so much, Xiuxia, for the very nice introduction. And I learned a lot from Oliver's talk. So I will share my screen. Hi, everyone. Thank you for attending today's webinar. And so today I'm going to talk about the ADP informatics for untargeted mass spectrometry-based exposomics. As Xiuxia said, I'm part of the NC Here Hub. So this work has been done in very close collaboration with all the labs in the NC Here Hub, also with Shankar's lab at UCSD. And also we thank all the funding support to make this work possible. And so we all know now on the science horizon, discovery science is taking a foothold now because of the wonderful instruments that our community has access to. And so based on the Wikipedia definition, the discovery science aims to find new patterns, correlations, and form new hypotheses through analyzing large-scale experimental data. So compared to the traditional hypothesis-driven science, so discovery science is really to try to observe our nature, and in contrast, the hypothesis-driven science tries to explain nature. So the nature of the discovery science determined that it's very heavily data-driven. So it's very, very important for advancing the relatively new field, which is exposomics. There are so many things we don't know yet. When it comes to exposomics, LC and GC mass spectrometry-based methods have been widely used for analyzing samples from exposure studies. The data is complex. We have seen from Oliver's talk, not only complex, but also the scale of the data is huge. So this means there need to be dedicated informatics tools for making sense of the data. And my style has made tremendous contribution to the field, and now we see LC beam-based on the horizon. And as part of the NC Here Hub, my group has been trying to address some of the informatics issues. So overall, the informatics challenges in exposomics can be put into one of the following three categories. One is to pre-process the data, which means to extract compound signals from the raw LCMS or GCMS data. The second category of challenges is how to identify, annotate, and predict the compounds. And we have seen Oliver's talk on the many environmental compounds that he has identified. But how about those compounds that have not been identified? And we need to annotate them, provide some pointers, and also to predict them to see how real they are. Eventually, we have to use experimental method to identify them, of course. And finally, how to put the data into a biological context, give it a reasonable biological interpretation. So these three categories of informatic challenges is in their particular position in this workflow. We have raw data coming in, pre-processed data, for the relative quantitation data, we do statistical machine learning analysis. And for the qualitative information, including accurate mass, retention time, and fragmentation spectra, we try to identify, annotate, or predict the compounds. And then finally, all the information will hopefully help with explain some biology in the data. So my group has been trying to develop informatics tools to tackle these challenges. So again, in this workflow, we have developed ADAP Big for data preprocessing, and users can access it at this website. And for discovering patterns and discovering related compounds, we are developing ADAP Analytics. And for compound identification, annotation, and prioritization, we have been developing a web resource called ADAP KDB. And for biological interpretation, especially for foodome, and also foodome is also considered as part of the exposome, we have been developing web resources to tackle that. And obviously, we don't have time to look into detail each of this, so we will look a little bit closely into ADAP Big, ADAP KDB, and the multiple food. And ADAP, we started with developing data preprocessing, and this has been available through MZMine for a number of years. And for those of you who are interested, you can read the tutorial and give it a try. And the development of our ADAP ecosystem has been ongoing for more than a decade. Here I'm showing the past work in developing the algorithm. And for the software development, you can see we have gone through a few iterations from R to Java to, and also has Python versions as well. So the most recent development is ADAP Big. And this is a Java-based software tool. This is really the user-facing software tool. And it's freely available on the ADP-Big website. And I'm going to talk a little bit more about this. And of course, this work has been developed by a team of programmers in my group, especially Alex. Here I highlighted a little bit in developing our ADP-Big. So here I'm showing the specific computational steps in the ADP-Big workflow. And we do the traditional way still, even though we're still developing better approaches. We detect the peaks, align those peaks, and adduct annotation has to be done. And when we have large-scale studies, and ADP-Big was specifically developed to handle large-scale studies, and we have to do batch effect correction and also make the software capable of handling big data. ADP-Big has installation packages for both Windows and Mac. And here I'm showing a little bit more details. In order to make ADP-Big capable of large-scale analysis, Professor Oliver Fiehn has presented the bin-based approach using AWS. Here, ADP-Big was originally designed for a desktop application because we consider uploading data to cloud could take time for open source software. But hopefully this will provide an alternative solution for those of you who have big data. And visualization is critical in the development so that users could interact with your data closely. And this can be deployed either on a desktop computer or of high performance computing cluster. So this means if you have AWS accounts, you theoretically can also deploy this on a cluster environment. And of course, we have been trying to rigorously evaluate the performance of ADP-Big. The first evaluation, of course, is to look at the overall relative quantitation. And here we have been comparing ADP-Big against two commercial software tools, which is Progenesis and Compound Discoverer. They are from Nonlinear Dynamics and Thermal, respectively. Here we used the pilot project data that were produced in the past couple of years from the HERE program. Here we see the PCA plots. This is from ADP-Big, this is from Progenesis, this is from Compound Discoverer. And you can see at the very high level, the relative quantitation produced pretty similar PCA plots. So this is very high level. So from relative quantitation point of view, we could say the three software tools performed comparably. And for peak features, these are just the peak features. Many of them might even not have corresponding MSMS. We wanted to take a look at how these three softwares compare. This is still an ongoing comparison. We have not completely finished it yet. And as we all know, there are usually many parameters when we do data preprocessing. So many of those preprocessing parameters would influence the final comparison. So we are still trying to make sure we choose the best possible parameters for each of the software too. What you are looking at here are some preliminary results, and we are still refining our analysis. So here this Venn diagram shows the number of peak features that have been detected by the three software tools. And for the peak intensity patterns, we have observed some of the peaks if they have been detected by both ADP and Progenesis. For example, here their intensity pattern is remarkably similar. But for some peaks that have been detected by both software tools, the peak intensity pattern, you see there's no correlation whatsoever. So we can see there are still differences. And we are still doing some very detailed analysis to look into this. How about in terms of compound identification? This means to match against your in-house library. Your in-house library has been produced using the same analytical platform. So here we are talking about results for data that have been acquired in Dr. Susan Sumner's lab. Her lab has developed an ontology system to provide evidence basis for compound identifications. And for matching against in-house library, there are 3 levels, OL1, OL2A, OL2B. Here I'm showing the criteria to be assigned to be to that particular level, and also for compounds that have been matched against public databases. Today, we are not going to talk anything about the PD level, the public database annotations. We'll only look at match results from after matching against the in-house library. So OL1 is the highest level, which includes matching in terms of mass, retention time, and MS. So this is the comparison between ADP and progenesis at the highest level. You can see there are some differences still. And if we add OL2A level to this picture, we can see the overlap drastically increases. If we also include OL2B, so basically we consider all the OL level, the overlap is quite substantial. And ADP shows, this also shows ADP was able to detect some more compounds at the OL level. That's what we want to do for exposomics, because some of the signals could be not that strong. So a big test for our informatics is, can we robustly and confidently detect relatively weak signals and still be able to identify them? So with that comparison, that give us some confidence in applying ADP big. So this is one large-scale study that we have carried out. This is actually carried out before our rigorous comparison, but still we have seen good results from this study. And here is a large-scale study with more than 3,000 plasma samples, and they were distributed into 42 analytical batches. So ADP big was able to handle it. And for exporting the results, we export the results after matching against in-house reference library, also matching against public libraries. Here I am showing screenshot of the results after matching against NIST and HMDB. Okay. So that's ADP big for pre-processing. Of course, ADP big extracts MS and match them to MS1 peak. And the compound identification and annotation actually is done not in ADP big, it's done in ADP KDB, which is a web resource. And as we all know, for exposomics, compound identification and annotation is even a bigger issue than metabolomics, because there are so many compounds are unknown, even there are many unknowns. The library is still limited. And so how can we identify compounds, annotate compounds, and predict those unknowns, especially unknowns? And this is what ADP KDB has been designed for. We have not, of course, completed all the tasks. This is still ongoing development, but we are hoping to continuously making progress. So in terms of compound identification and annotation, there are 2 very big challenges. One is it's very time consuming to match against the library and also reviewing those results. Despite advances in library searching, but still human reviewing is still necessary to provide highest level confidence in the final results. And another challenge is exposome related spectral and compound libraries are scattered in many different places. And so in order to tackle these 2 challenges, what we have done is to try to speed up library search within ADP KDB, also develop user-friendly visualization features to make it easier for human to review the results. And also we have been trying to assemble exposure library, compound libraries to put into ADP KDB so that we can do the search in one place. Here I'm showing the landing page of ADP KDB. Within ADP KDB, you can build your own private user account. And if you trust us, you can put your own library under your private account to do compound identification. And you can also use ADP KDB to search against public libraries for compound annotation. And there is simple search and batch search features available. And of course, you have the search parameters under your control to control your search. And you can also visualize, sort, and filter searching results from the GUI. And also, ADP-KDP has another feature, which is consensus spectra. The idea behind this, in terms of the relevance to exposure studies, is because there are the library is still limited, but there is huge amount of data in the public domain. For example, metabolomics workbench in North America, metabolites in Europe, for example. So one idea behind that work is, can we use those data to do something? And so that's what we have done to create a library or consensus spectra so that you can search against as well. If time permits, we'll talk a little bit more, but right now let's talk about the basic things first. So within ADP-KDP, you can do library search, and you can search GCMS data, LCMS data, LCMS data. And for your search, you could specify your own search parameters. In terms of libraries, in relevance again to exposomics, under the private here account, here you see the list of libraries that our here core could search against. And there is still a bug in our library right now. You can see ToxCast repeated several times, but there's no bug, I don't think so, in terms of searching the library. So here you can see the environmentally relevant compounds in the here common core, GCMS core, here common core, LCMS, and the Sumner in-house library has a big set of environmentally relevant compounds. And there is the multi-class panel also from the HERE program that's from a different lab within the HERE hub, and compounds in drug bank, and also the high quality NIST MS spectra for both positive and negative. And also we have some environmental compounds that have been detected at the Duke HERE hub. So all this is available for search and within the HERE hub, environmentally relevant compounds. So after you search your public libraries and the private in-house libraries, and we really took efforts to develop visualization capabilities to make it easy for you to examine your search results. Here you can see the spectral plot and the detailed information about the match. And you can do filtering, you can sort the results, and really we try to make it easy for you to explore the searching results. So that's a compound search engine with the ADP-KDP. How about for those compounds that are unknown, either known unknowns or unknown unknowns? They are just not in the library. So what can we do about them? Obviously, we want to eventually identify them, but there are so many, as Oliver mentioned earlier, maybe for about 1,000 signals that could be matched against library, more than 8,000 of them actually could not be matched. They are still unknown. So in order to tackle that issue, we had one specific objective in mind, which is to leverage the vast amount of metabolomics data and exposomics data in the public domain and provide information to help prioritize those unknowns for very costly compound identification, and also hopefully help with compound prediction. This is still ongoing work. But how do we achieve that particular objective? Step number one is to use the same software to pre-process raw LCMS and GCMS data. Hopefully that will make it relatively easier to harmonize the data across labs, and also extract spectra from the raw data for various compounds, build a library, a consensus spectra. If this compound has been observed over and over again from different studies, that speaks something about the spectra. And so we build the consensus spectral library and also make the spectral library searchable, and also compute study-specific statistical significance. And that's going to be used for us to prioritize those compounds. Okay, so let's look a little bit more in detail. I might not be able to finish. Let's see. So here you see in ADP-KDP the list of public available studies that we have processed. And for a particular study, for example, this is from metabolomics workbench. If you click that, it will take you to another page to show all the compounds, either known or unknown, that we have determined, and all the spectra are here. For a particular compound here, for example, if you click, that will bring up the spectrum, what the spectrum looks like, and more details. And also, if you want to learn more about that particular study, we provide the URL for you to jump to Metabolomic Workbench and to see more information. And coming back to ADP-KDB, if you click Files and Users, you could download the spectrum that we have already collected, and you could look at the data and search against them using ADP-KDB or another search engine that you like. And the library of consensus spectra that have been built from all these studies and could be examined. If you are in ADP-KDB, click View All Consensus Spectra, you can see the list of all the consensus spectra, and click on a particular spectrum. Here, you can see more information for that particular cluster of consensus spectra. Here you will see from what studies that the underlying spectrum has been extracted. So this is mostly from Metabolomic Workbench. And also you can see the distribution of diseases and multiple different metadata. I'm only showing diseases and species within that cluster. And this gives you some information to help you determine whether or not to go after to try to identify those unknowns using experimental methods. And there are more there that we don't have time, so you're welcome to explore. And for the library consensus spectra, you can filter them based on species, sample sources, and diseases as well to narrow down to things that you are more interested in. And as I said earlier, this library of consensus spectra could be searched against. This is ADP-KDB consensus spectra, in addition to other publicly available spectra that you could search against. After the search, you again could examine, filter, and download the search results. And in addition to go from ADP-KDB to Metabolomics Workbench, users could also go the other way from the Metabolomic Workbench coming to ADP-KDB. And I just found out this morning, actually this link is not working. So it used to work, so we need to look into that. This is still ongoing work, and we welcome your test it and give us feedback. So that is ADP-KDB. The final piece is our metabo food. The idea here is try to link food to metabolic pathways to diseases. This is, of course, very relevant to exposomics because what we eat is considered part of the exposome. And so we have been developing this web resource and to help people to explore the complex relationships between food and diseases. And here I'm showing the hierarchical pathway related to particular foods. And this slide is a very high level overview. And the way we have extracted information is through this ongoing development of our workflow to try to extract information. The library we have been using, including HMDB, CAG, Reactome, CTD, and more databases we hope to bring into the picture to try to harmonize data across different databases. And one centerpiece of this work is the Sankey diagram to visually explore how food affects our health. And with that, I'm going to stop here and take your questions.
Thank you, Xiuxia, for that very interesting question. I'll remind people again, you can either type in or you can raise hand. I think we have a raise hand feature. You can ask directly. Oliver, do you have a question? Or
Sure.
I didn't mean to put you. I saw you unmute.
No, I just wanted to be here. But as I don't see questions in the Q&A, I might ask Xiuxia directly to Dr. Du. That's great. I saw that there are in-house libraries. Why are they in-house and when will they be published?
Oh, this in-house is Susan Sumner's library. And I think I have heard she talking about to make it public available. And right now, in ADP KDB is still behind the firewall right now. Yeah, that's the in-house library.
Yeah, I don't think we should have in-house libraries.
And I have a question, because this is kind of related to an earlier question. A person, an audience asked, how much is unknown, how much is known? In the ADAP KDB, the knowledge database for the consensus spectra, you mentioned roughly some numbers. I know this is ongoing work, things can change as you, get data from more studies, etc. It just, as of now, how many are you able to, what kind of percentage you can match to known and what percentage you cannot? And for the ones you can match to known, how many are environmental chemicals?
Okay, I don't have the specific statistics, and so qualitatively speaking, it's similar to what Oliver said earlier. A very small percentage of the signals could be matched against libraries, including both in-house and the public library, because the library is still small, even though we combine public available libraries. The percentage of signals that could not be matched with anything is still the majority. And the environment, in terms of environmental compounds, that of course is determined by how many compounds are in the in-house library for confident identifications. I don't know the specific number. I think it's also very depends on the type of samples that's being analyzed. I think Yan is here. What's the percentage of environmental compounds in some of the studies? I think you know more about that level of specifics.
That's fine. That's already a good understanding.
Okay.
I have another question actually to both speakers. So you can both introduce, how the data processed in the lab. I think, I get really excited. I feel like you should talk to more people so people can use. But in the same time, I have a question for the kind of the users who don't develop their own computational workflow.
Yes.
How do they choose is one thing. The other thing is for, those who develop those pipelines and computational workflows kind of moving forward, because Xiuxia mentioned that, using the same software process raw data. So how the field should be moving and any insight will be helpful. Thank you.
Yes, so we have written an R01 that is being reviewed in 3 weeks. In case you're a reviewer, please be mild. I think I think we need a national repository, an actual repository where people upload the data and get processed. It should be harmonized and standardized, and it should not be, like distributed and everybody does the same what other people have done before, trying to kind of match and merge. It should be, with internal standards. It should be very much like genomics is done. And I think we are on the right track. I mean, Dr. Du has shown that her workflow gave very comparable data sets, results when using different software. So that's benchmarking, that's perfect. That was well done, Xiuxia. And, that's, I think, how the nation should go forward. I think the approach that the Metabolomics Workbench was doing over the last 10 years is wrong. By just getting process data and uploading them, I think it has to be done based on a unified and harmonized data processing workflow.
Xiuxia, do you have anything to add?
On the same note, there are lots of data in Metabolomics Workbench, and I'm completely on the same page as Oliver. I think it needs to be processed in a uniform fashion. The biggest challenge that I have been facing when trying to harmonize data in the public domain is the in-house library is not there. So the best I could do is to match the signals that I have extracted against the public libraries. but we know that the confidence is definitely cannot compare with if I could have matched against the in-house library. So that's my probably the biggest challenge in trying to harmonize data. And another thing is also to second what Oliver said is, again, for harmonizing data, we have to align data sets across labs. The community does not have a decent set of internal standards. Some labs might have one or just a few, and that even that level of information is even not provided in the public domain. So aligning the compounds could be problematic without a decent set of internal standards. So I guess hopefully through this platform, Oliver and I could both advocate.
Yes, it's not just the platform. We also need to convince people to harmonize data acquisition methods. So it can only be aligned if people use the same methods and the same internal standards. That, is possible. I think many laboratories are open for that. Many laboratories don't need to use this specific C18 or that specific C18 column. They wouldn't mind use something else. But this needs to be a concerted effort by also funding agencies. So I think the time is mature. We are ready. Through the last 10 years of the Metabolomics Consortium, we have a good community of people who have shown that they can work together, also through the other consortiums like the Exposome and the food and similar initiatives. I think as a community, we are ready to work together.
Thank you both so much for the insights. Hopefully, we'll have more conversations in other places, society meetings, all of those. So we are a couple of minutes past the hour, and thank you both so much for the wonderful presentation, and thank you everyone for joining. Our next seminar is coming up soon. Okay, I think a slide is okay. Here you go. Please stay tuned. The announcement will be out soon. Again, thank you everyone for joining, and thank you for our speakers. Bye.